thank you all very much for joining the Jetson March open call. As usual, we have two excellent talks for you. And the first of those is going to be Jordan Toullec, hopefully I pronounced that right, who's going to be talking to us about patterns in the carbonate pump. So, Jordan, over to you. Thank you.
Thank you, Adrian. I'm sharing my screen. Okay, tell me if everyone sees my screen.
That's great. Off you go. Thank you.
Cool. I will start. Okay, so hello everyone. So thank you for joining for this monthly open Zoom call. I'm Jordan Toullec, and I'm from Ghent University in Belgium. And today I will talk about my ongoing work on the spatial and seasonal patterns of the oceanic carbonate pump. So it's a combining study between particulate inorganic carbon flux and remote sensing observation, so thanks satellite. First of all, I want to say a few words about biological carbon pump, even if I think almost everyone is very familiar with this process. But it's also a nice opportunity to me to present my own drawing and sketch. So first, as you know, phytoplankton, thanks to photosynthesis, contribute to uptake CO2 from atmosphere. So via this equation, so they convert inorganic carbon, so CO2, into biomass, so organic carbon. And this phytoplankton could then be transferred into higher trophic levels, such as zooplankton and fishes. But it can also sink through the water column via the aggregate formation and also the fecal pellets emission from zooplankton and fish. So as you can see on the different picture, it's different type of phytoplankton aggregates and also fecal pellets. So some phytoplankton, such as coccolithophores, have the capacity to calcify. So they can produce calcium carbonate, which is also called PIC, thanks to this process, so the calcification. And as you can see, this calcification process could release CO2 in the atmosphere. So that is why we call this process the counterpump effect. And of course, when you have a aggregate formation, you also contribute to export deeper and deeper those particles, which is also composed of carbon, of course. So now, during the transfer of calcium carbonate through the mesopelagic layers, you could also have dissolution. So this is this process, and dissolution contribute to fix CO2 into alkalinity, and also contribute to decrease the PIC. Coccolithophores are one of the main primary calcifier in the ocean, so they are the most important contributor. And the interesting aspect of those algae is that they are composed of both inorganic carbon, so the calcite, and organic carbon, which is composed of cells, so all the nucleus, cytoplasm, and different carbon pools. And basically, the inorganic carbon composed of calcium carbonate are commonly called coccolites, and those coccolites are like a tiny shells all around the cell. So you can see some example on this picture. So it's different coccolites from different coccolithophore species that I have in culture in my lab. So as you can see, you have different size, different shape, and they and also some specific shape. So that is this case. And the interesting fact from this is that during coccolithophores bloom event, for example, they constantly produce a lot of these coccolites, and they release free coccolites in the seawater constantly. So sometimes even during some blooms, we can have like more than 200 times more free coccolites than cocosphere in the seawater. These particles have a specific and unique light scattering properties because they are birefringent materials, calcite. And based on the ocean color remote sensing, we can basically evaluate the PIC stock and production thanks to ocean colors. So this is the map you see here. So it's the particular inorganic carbon surface concentration at global scale, and it's also a mean annual concentration. Basically, you see that you have a lot of PIC in Southern Ocean, in polar ecosystem, and also in North Atlantic. And we also have to note that we observe a high PIC concentration close to the continental margin. And that is the case, for example, in North Sea. So it could be probably a bias of the continental plateau and particular resuspension. In my work, I'm based on a satellite product to estimate PIC production at global scale. So for this, I use a model adapted from Hopkins and Balch from 2018 from a paper. And inside this model, so I need the sea surface PIC concentration based on remote sensing. I also need some experimental growth rate constant from Emilianux because Emilianux is one of the most important species of calcifier in the ocean. So basically, they produce the majority of the big cocoetophore blooms in North Atlantic and also in the Southern Ocean. So this is why it's important to fix some constant inside our models based on the experimental study. So we will still need experimental work for build large models. Inside this model, I also use the sea surface irradiance, so the light intensity, the sea surface euphotic zone depths, so the depths as where the light could go and could still induce photosynthesis. And of course, I also need a sea surface temperature because a lot of physiological parameters, like such as growth rate and nutrient uptakes are determined by the sea surface temperature. By combining all of this, I can get a monthly PIC production estimation, so according to different months. And as you can see on this small animation, you have different patterns during the North summer and South summer, so corresponding to the North Hemisphere summer. In my work, I also use a huge data set collection. So based on the sediment trap data, so I think everyone knows what is a sediment trap. So I'm using PIC flux, which has been collected during almost 3 decades. So from 1992 to 2009, and it's about more than 6,000 observations. And in my data collection, I only use monthly sediment trap deployment because my idea is just to do a matchup between the monthly satellite data and the monthly sediment trap PIC flux observation. The map here represents all the data I got in my data set. The white triangle corresponds to monthly deployed sediment trap, and the yellow circles correspond to thorium-derived fluxes, which is quite different, but it's still good to estimate short-term PIC flux. And also important to note that the different colors in this map correspond to different biogeochemical region, so defined by the RECAP2 region, so the regional carbon cycle assessment and processes. And in my work, so I'm just trying to compare the different regions each other to see if you have like a common patterns and if this region could be specifically linked with a specific biogeochemical patterns. My question for this ongoing work is the following one. So can we link PIC production in the euphotic zone and deep PIC flux? On these two maps, it's annual euphotic zone PIC production on the top and euphotic PIC standing stock on the south. So based on remote sensing. And so what the first thing that you see is that we have an opposite pattern between the production and flux. So basically, if you look at the standing stock, we have much more PIC stock in the Southern Ocean and also in the North Hemisphere, close to polar region, and less in subtropical gyre and also in equatorial upwelling. And that is absolutely the reverse pattern with PIC production. So because it's based on my models, and of course we have much more higher temperature, we could expect also higher physiological processes and also a bigger growth rate. So now if you combine these two maps, we can get the euphotic zone PIC residence time, which should look like this. So basically what we observe, we have a higher residence time in high latitudes, such as in Southern Ocean and also in North Atlantic. This is also the case in North Pacific. So basically the residence time corresponds to the time where your particles could just stay in the water column. On this map now, it's the different seasonality of PIC production according to all the different regions. So the box plot corresponding to the PIC flux in the euphotic zone in petagram carbon per year, based on the monthly climatology. And this monthly climatology has been based on 3 decades of PIC production models. So I did my model for 3 decades to get like a global mean and median and equartip for the monthly climatology of PIC production. So what we can see, for example, in Arctic and North Pacific and North Atlantic subarctic, we observe a very nice seasonality. So we characterized by higher PIC production during spring and summer. So it's okay. Now, if you go down, we observe less seasonal patterns, except in North Atlantic, where we still have higher PIC production during May to June. If you go to the equatorial upwelling, so it's not really seasonal. In South Hemisphere, we have the reverse pattern of the North Hemisphere, because basically it's the opposite phenological process, because it's now during the North Hemisphere summer months, it's corresponding to the South Hemisphere winter. It's also notable for subantarctic and Antarctic. In this graph, it's a summary of all the different seasonal patterns of PIC production monthly climatology for all the different regions. And if I plot the sediment trap data in both mesopelagic layer in blue and bathypelagic layer in black, we don't observe that much nice seasonal patterns, except if sometimes, for example, if you look at the Arctic, we have, of course, higher sinking PIC flux for both mesopelagic layer and bathypelagic layer during July to September, where we have the pulse of PIC production. We observe the reverse pattern for Antarctica with a lower PIC flux during the winter months, South Hemisphere winter months. When we look at the other, we don't see regular patterns. It's also important to consider that my PIC production model is based on only co-cultivar growth rate, because it's based on remote sensing. But we have also different other contributors for this flux, for the flux based on sediment traps, so such as foraminifera and theropods, which they are not counted inside my models, but they still exist. So on these 2 different maps, so it's a work from a publication of last year. So it's an estimation of the total carbon biomass, the biomass of both theropod and foraminifera in the global ocean. So this is the annual mean of an output model to estimate the relative biomass of these 2 components. So what we can see, so for example, for theropod, it seems that we have a higher biomass close to the upwelling region and also in polar ecosystem. And for foraminifera, it's mostly at annual mean, it's mostly in North Atlantic. When we look at so it's a log 10 scale. When we look at the biomass, we also observe that the magnitude is very different between both. So we have very much less foraminifera than theropod. So what I did with that, so I also tried to observe the seasonality of these 2 organisms. And I did a stack in terms of PIC standing stock for co-cultivar in green, theropod in purple. And foraminifera, we cannot see them because they are very small. So we can just picture the little red line on North Atlantic. But the thing is, we look at this. So when we look at this, we observe that we have mostly Cocolithophore. Cocolithophore dominates the standing stock, except in equatorial Atlantic, where we have a higher contribution of theropod to the stock. But if we decompose these different taxa, for example, for theropod, we still see the seasonality in North Hemisphere. So that is the case of North Pacific, Arctic, and North Atlantic. And in the south, for Antarctic and subantarctic, we observe that we have less standing stock, PIC standing stock during winter months. This is also the case for foraminifera. But the important here is to note that even if we have like a quite high contribution of standing stock for a theropod, they would have like a much more higher residence time, and also they will have a lower productivity. So it means that I don't think it is an interference inside my model of PIC production. So now, if you look at more the sediment trap data, on this box plot, so it's all the different bioregion in y-axis, and on the x-axis is the PIC flux, considering the export flux, the export layer, the mesopelagic layer, and above the mesopelagic layer, so the bathypelagic layers. And what we observe that for the orange box plot, so the export flux, they are always higher than the mesopelagic and bathypelagic layer. So we don't observe that much difference, obviously any significant difference between mesopelagic flux and bathypelagic flux. So it means something interesting, that we have a big loss of PIC above the mesopelagic layer, so represented by this decrease of flux. And this thing is supported by many studies. So for example, if I take this paper from Olivier Sulpice from 2021, at a global scale and annual scale, we observe that we have a massive pelagic dissolution in the mesopelagic layers. So it's a way above the lysocline. So the lysocline is the depth where you chemically supposed to start to dissolve the aragonite and calcite, which are calcium carbonate. So it means that what we observe here, so this massive pelagic dissolution is caused by biological activity such as grazing and microbial activity. So my strategy in my work has been to do a comparison between PIC production based on my model and the flux regarding the seasonal variation. One big aspect of my work is even if we have a large dataset collected during 3 decades, only 75% of my data from sediment trap have been deployed before the satellite covering, because the first satellite products started in 1996. So it means that if you want to do a perfect matchup between the year and the months and a specific location, the subset will be very, very, very low, and we will lose a lot of points. So we decided to work with monthly climatology. So as you can see, if I take an example of North Atlantic, subarctic, so on this time series, you have the PIC production in petagram carbon per year that I performed over 3 decades, from 1996 to last year, to last December. So we observe we have a constant cycle of PIC production, basically higher PIC during spring and summer. And if I do a mean of this 27 years, I've got this box plot distribution. So the y-axis is the month, so January to December. And we have this quite nice seasonal pattern. So I use this climatology to do a matchup with my sediment trap data collection. If we still stay in North Atlantic, so we observe, so every yellow dot are sediment trap data deployment, and we observe that it's nicely spread all over the North Atlantic. But when we look at the monthly climatology PIC production, estimated based on my model, on the models, So the x-axis is the different months of the years, and the y-axis is the latitude. So we observe we have a latitudinal variation of PIC production. So basically, if you compare West Atlantic basin, south of Iceland and Norwegian Sea, we observe a delay of PIC production, and also in terms of magnitudes. So it means that we have a less PIC production in Norwegian Sea, for example, and it occurs mostly during July and August. So here another example of my work. So in this graph you have a time series from 1999 to 1990 of in black. This is the PIC production based on sediment on satellite, so mostly climatology, and the red are the PIC flux from sediment trap. So what you see, it's we have a nice matchup. So based on this, it's possible to do linear regression at this layer. So it's one example because it's the best. And if I do this for all the layers, so I get different linear regression for 6. I decided to work with 6 different layers of depths. So from 100 to 500 meters to more than 4,000 meter depths. And I repeated this for all the different sediment traps for every region. And I got a statistic based on that. So on this graph, I will show you how the relation between PIC production and flux variates between the different regions. So basically what I will show, so when we will have red colors, it means that we would have like a positive linear slope, meaning that we increase the PIC flux when euphotic zone PIC production increase. And when it's blue, it means that we have a negative linear slope, meaning that when PIC flux decrease, the euphotic zone PIC production also decrease. So it probably means that we have a dissolution process occurring in water column. So I've got this. So first thing, so we observe that in North Atlantic subarctic, we always have red colors, meaning that we have a positive relation between euphotic zone PIC production and flux. And then also we observe that it's very patchy between different parts of the ocean. So when it's white is because we don't have data. So it's we don't have data. And when I put hatches, it's when I didn't find any significant correlation between both variables. So it's also important to note that, for example, if you look at only the North Atlantic, so we have a switch of the linear slope between North Atlantic subarctic and North subtropic Atlantic. So it means that we have different processes, opposite processes between these two areas. So if we get more into the statistic, so this new map corresponding to the R square, so basically the powerful of the linear regression. And so you observe that first that the R square is quite low for all the region, but it is quite high, we can say, for North Atlantic. So it means that the linear regression I did was much more relevant in those regions. So in terms of process, so I have few hypotheses of what we observe this pattern. So the first could be the shape and the driver of the export. So this two map has been based on the data from Nowick et al. 2022. So it's a relative contribution of fecal pellets to the export and the relative contribution to aggregates to the export. So for fecal pellets, we observe that we have higher contribution to flux in subtropic compared to polar region and higher latitudes. And so we know that this region are driven by mostly small zooplankton grazer, such as microzooplankton and small copepods. And those microzooplankton and mesozooplankton also put a grazing pressure all around the year. So it means that we don't have seasonality of grazer. It's much more constant during the year. It's also important to note that we have some evidence that microzooplankton, such as Oxyrhis marina, so very small organism, could dissolve PIC. So as you can see on this work from Ancia et al. 2008, it has been observed that copepods don't necessarily induce the solution. So this is the fecal pellets, and we observe like almost intact coccolith inside the fecal pellets. But concerning the vacuole from Oxyris marina, we observe dissolution of calcite. If I look at the aggregates, so we observe that we are mostly aggregates contribution to carbon exports in higher latitude. So it means that it's also important to note that we have much more seasonality in this region, meaning that we have basically when we start to have the bloom event, so the spring bloom, so we increase a lot the biomass, and at the end of this bloom event, we start to form the aggregates. Grazer come and start to graze and produce fecal pellets. So it means that the aggregates flux is very located in a short period. On this sketch, it's a summary of my hypothesis. So in the case where we have like a seasonal pattern, so a seasonal grazing mostly done by large zooplankton such as large copepod, if they graze on a different type of phytoplankton and also calcifier phytoplankton such as coccolithophores, I think we can expect to an integration of coccoliths inside more or less large aggregates, and this integration contribute to first produce a ballast effect and also conserve the calcite from coccoliths inside the aggregates. But it could also be the case for fecal pellets of copepods, because we also have some evidence that they can just conserve PIC inside the fecal pellets, so via the peritrophic membrane. So now if you just compare to a continuous grazing through the year, so that is the case of a subtropic ecosystem. So mostly where the grazing is mostly driven by microzooplankton and small copepod, we could expect much more dissolution in the surface, in the euphotic zone. So if you consider the microzooplankton vacuole dissolution, and also even if the sinking flux is mostly driven by fecal pellets, we can't expect much PIC inclusion inside the fecal pellets. So here is a picture of one fecal pellet from copepods with the evidence of calcite inside. So it's this band of being based on microscopy with polarized light, which reveal the birefringent materials such as calcite, and we see that we have a lot of spots of birefringent material which are coccoliths. So as a take-home message for this presentation, so first we observe that the euphotic zone PIC production and deep sinking flux could be very patchy in the ocean, but they are positively correlated at a high latitude. Secondly, so the phenology of phytoplankton and also zooplankton grazing pressure could potentially induce PIC conservation or dissolution. So this means that probably the pattern of this high dissolution of PIC in the euphotic zone and in the mesopelagic layer is probably due to complex ecological dynamics which integrated different zooplankton taxa and also different functional traits. The PIC time residence is also important to this flux patterns. Also the aggregates and fecal pellets repackaging, probably. And also the type of grazer. So now, based on this, these findings can be used to create a remote sensing-based models, because my analysis has been done on mostly climatology of PIC production. And now my next step is to produce a remote sensing-based model to predict monthly climatology of PIC flux at different depths. So on this graph, it's the first result I got. So it's in x-axis the different months of the years, and in y-axis is my predicted PIC flux for different depths. Okay, so it's done. Thank you for your attention, and if you have questions, please come on.
Excellent. Thank you very much. Fascinating stuff. We're a little pressed for time, so maybe if we have one quick question, if somebody's got a quick question. Yibin, did you want to ask your question? maybe I can read it out.
I can read it too. So
Okay.
very nice talk. I probably missed something during the talk. Can you remind me how you compute the PIC export flux at the different horizon based on the regional average value? Yes, the following are based on from sediment trap dataset or remotely sensed PIC production multiplied by the export ratio. I didn't use the export ratio so far. So if you mention the graph I show, it was only the PIC production and not the flux. So basically, we can it's stuck. Okay, my computer is stuck. Oh, it's annoying. No, it's okay. Yeah, so basically it was the PIC production based on remote sensing, but only in the euphotic zone, so at the first meter of the surface ocean. And my idea is to link actually this euphotic zone PIC production and deeper flux. And the data I put on the graph, so it was bar plots with dot plot on it. So this one. So what I did, so it's I just did a mean. So the different point corresponding to the mean of the deep PIC flux month by month. So we see that we have a pretty nice coverage of all of this dot. But for example, in South Indian Ocean and Subantarctic, we don't have data. But sometimes we just also have just one data. So this thing is not really representative of the system because it's we have a lot of hole and of course we have a latitudinal variation of and range of PIC flux. So this is why I decided to just aggregate them into a different region.
Okay, thank you. I'm afraid we're going to have to sort of move on.
Yes, of course.
So
thank you
once again for your talk. And say if anybody would like to ask Jordan questions, I'm sure he'd be happy to receive them by email.